right. And then take it away, Annemarie.
So thank you, Laura, for your introduction, and thank you for the invitation. It would have been nicer to be in Leibniz, really, but this will do as well. As you said, I will be talking about monitoring attentional engagement through interpersonal physiological synchrony. But first I will tell a little bit about the research organization that I work at, TNO. So TNO is a bit like Fraunhofer, I would say, or as your own institute. It's a little bit in between university and really applied research, or it is applied research. And there are different units in our organization describing different application areas as depicted in this picture. Do you see my arrow, actually? Yes? Good. There are different locations of TNO across the Netherlands, and we are here in Soesterberg in the center of the Netherlands. And it's really the human factors part of TNO. We do human factors research. And here are some pictures of our facilities. So there is a telepresence lab. There's a big machine that can throw you around to simulate air fighter pilot movements, for instance. And of course, there's a physiology lab. So my research is on mental state monitoring. So monitoring emotional or cognitive states using physiological and other types of signals. So I also wrote some fields here. It's close to affective computing, passive brain-computer interfaces, or neuroergonomics. That is the area that I work in. And we really try to combine different fields. So we have traditional neuroscience, where there are really good experiments being done that inform us about the signals that go with certain psychological processes. But also there are these new methods and equipment that allow us to take that research out of the lab, to go from these very controlled settings to more real life circumstances. We have wearables that we can use to monitor physiological signals, and we have different computational methods like machine learning that is being applied on neuroscience signals, so that it is now possible to interpret brain signals, for instance, for one person and at one moment in time, rather than having to average a lot of signals across a lot of participants and a lot of trials. So this really enabled new types of research and applications. Also, of course, we have more data capacity and computing power, all enabling new types of work. And then this third aspect is the application. So maybe we can indeed say something about mental states from brain signals or other signals, but why would you want that? In which applications does that have added value? And can you do that in real life situations where there's a lot of noise and other things going on? So that is really the intersection that we work on. Okay, so as I mentioned, we look at physiological measures as source of information about mental states. But why would you actually want to do that? Why don't you just ask people? So there are specific advantages of physiological measures. In principle, you can have continuous information on somebody's mental states, like attention, emotion, workload, instead of snapshots when you ask people. You don't have distraction or workload, if you have to ask people all the time, what are you feeling? What is your experience now? That adds another task to the task that the participant or that the user is doing. Also, verbal reports could be distorted. There can be response biases, or it can be distorting. So it can affect the experience or the workload or the emotion itself. And that is usually not what we want. So if we can monitor this mental state, what could we use it for? So broad categories of envisioned applications are evaluating systems or products in this continuous way, or even in a closed loop situation, you can make systems respond to the current mental state of a user. So adaptive automation, and therewith, for instance, improving the human-computer interaction in real time. So we want to go from lab to life, but there are really some hard challenges in this field. One of them is the ground truth mental state. So I was telling them, we want to know about people's emotion or workload or attention, but how can we validate whether we are correct? Because then that is not so straightforward, especially for instance, emotion. It is also often the models that we make are quite context specific. So they work in some contexts, but not in others, maybe. We also often want to relate physiological signals to events in the world, but we don't always have information about these events in the world. And intrusive sensors, so you don't want to stick EEG electrodes on you, preferably. But yeah, how can we deal with that? So a lot of people are working on these problems from a lot of different angles. The nice thing of the kind of feature or the interpersonal physiological synchrony that I will be telling you about in this talk is that it kind of circumvents a lot of these problems. Okay, so this physiological synchrony, we started working on that in 2018. So we had this big project, a 4-year project in Neurolab NL called Neurolab NL. That it was a kind of consortium in the Netherlands with different universities and institutes joining. It was about applied neuroscience for the benefit of youth, and with different strands. And we were in the strands wearables. And what we worked on was wearables in an educational setting. How may wearables benefit youth in an educational setting? And we zoomed into the idea of monitoring attention in the classroom. And the candidate marker of this attention was this physiological synchrony. So what is physiological synchrony? So that is the degree to which physiological measures of multiple people change in the same way. So we expect this to happen if people respond to some event in the world in the same way. And when does it happen? So if we all attend to something that is happening in the world, if we all attend to that, then probably our brain signals are similar, because the same events trigger the same response. That can be caused by shared attention or shared emotional arousal. So we kind of call that together attentional engagement. So we could use this physiological synchrony maybe to identify events that draw attention or are considered relevant. So if the physiological synchrony is high, then apparently something that the group thinks is relevant happens. And we can monitor the attention level in a group. So we got this idea from previous work that was done in EEG. So it was already shown that indeed moments of high physiological synchrony between viewers of a television series co-occurred with interesting events in that television series. Also, Dicker et al., Suzanne Dicker, they showed that physiological synchrony between students in a classroom predicted class engagement. So this was quite promising. Also, there's a lot of work on physiological synchrony in more the peripheral physiological field. So looking at heart rate, skin conductance. And there in that literature, it is often described not really as a measure of attentional engagement, but a measure of general connectedness. It's a bit more vague there what it means to be, to have the same, similar signals. It is, really discussed in terms of togetherness and these kind of terms. Also, there is a literature on synchrony in behavior. And of course, that can also, if you move in the same way, that can also cause synchrony in physiology. But we, and that can be interesting in itself, but we kind of see that more as a confound or too complicated. We try to keep that out of our experiments for this time. Okay, so what did we ask ourselves? Firstly, we were interested to move from EEG to peripheral physiology. So we saw already this previous research showed that synchrony in EEG is informative of shared attention, but can we also use easier to measure peripheral measures for that? Then can physiological synchrony really predict cognitive performance? And what if we measure it using wearables? So all of these questions, we looked at that in a lab experiment. Then we used existing data to see whether there's hope to also measure this physiological synchrony in real life school environments using wearables. We also touched upon the ethics of this. And after this Neuralab project, we also looked at other instances where we don't modulate attention by attentional instruction, what we did in the lab experiments, but where we expect attention to be modulated by a personal trait or by lack of sleep, and to see whether that measure is useful in that situation as well. And finally, we recently did an experiment when I did my inaugural lecture at the University of Radboud in Nijmegen. We also fitted the audience with some wearables to look at attention, whether people attended to my lecture. So I hope to get back to get at that in the end. Okay, the lab experiments. So what we did is that we presented all participants with exactly the same auditory stimulus, but we instructed participants to attend to different aspects of that stimulus. And then we compared modalities, EEG, EDA, so electrodermal activity or skin conductance, the sweating, more or less, heart rates, and we used both high-end equipment and wearables. So this is what the setup looks like. We presented people with an audiobook of 66 minutes, and we interspersed that audiobook. We just played certain tones or sounds through it. So firstly, emotional sounds from the IATS database. So it could be crying baby or laughing children or things like that. And also tones. a series of tones that some people had to attend to and others not. In the end, to also monitor how well they attended, we asked them questions about this audiobook, these sounds and the tones. So we instructed half of the participants to really attend to the audiobook, and the other half of the participants we instructed to attend to these short stimuli. So count the tones or really pay attention to the emotional stimuli. And then for all the modalities, EEG, EDA, and heart rate, we try to classify for each participant whether this participant was in the narrative attending group or the short stimulus attending group. And that was based on the highest synchrony that person had with this group or that group. So in this way, we could classify each participant and see how well that works. Also, we were interested because I already thought we had this audiobook and then we had these sounds. And we wondered, like, maybe the findings, if they are positive, they are driven by only these parts when these short stimuli were played. So we also looked at that. We zoomed in on the data around the short stimuli and like only the audiobook parts. Okay, very short, the methods. So for the physiological synchrony in EEG, we really exactly followed these other people's, Lucas Parra, Dmowkowski, and others who worked on this. So we did correlated components analysis, and we took the sum of correlations of the first 3 components. So that was kind of pre-processing. We don't look at the raw like signals, but this is a pre-processing step. For EDA and heart rate, we looked at Pearson correlations over 15 seconds running windows. So that is also a method that others used before. Oh yeah, and I have to say maybe for the EDA, we look at the phasic part of the skin conductors. Okay, so here are the results in percentages correct. So this is to what extent we could classify participants in the correct group. So 50% is chance, and clearly for EEG we could classify almost all participants correct. For EDA and heart rate, it also worked. So we were very happy with that. And also what was nice is that using the wearables did not decrease these percentages, even a bit higher. But yeah, we could use the wearables just as good. And yeah, that was also very nice. If we zoom in, so this was for the data looking at the entire 66 minutes of stimuli. If we zoom to the data around the beeps, or the effective sounds or narrative only, we could still do the classification. So that was especially maybe surprising or positively surprising for the narrative only part. And what we also saw was that each modality has its own specialty. So EEG is very good for these beeps, EDA for the effective sounds, and heart rate for narrative only. Okay, if we look at the attentional instruction and the performance, so the answers on the questions that we asked, then we see what we expected. So people really followed the instructions, apparently, what we also expected from the previous results that I showed you. But indeed, people who were asked to attend to the narrative answered more questions correct than the people who were asked to attend to the short stimuli about the narrative. And that short stimulus group better estimated the average number of beeps and remembered more effective sounds than the narrative attending group. So that was what was expected. But what is, of course, interesting, does physiological synchrony predict this performance? So what we did is that we rank ordered the participants in terms of performance for each set of questions about narrative, beeps, and effective sounds. And indeed, we found that a higher rank, so if you have more questions correct, you about the stimuli, for instance, you also have a higher physiological synchrony with the stimulus attending group. So we found that for EEG and heart rate. So really the degree of physiological synchrony is associated with answers correct. So it is kind of meaningful. It can predict cognitive performance. Okay, we also use this data to look at detecting relevant events. So, of course, in this case, we knew for some part when relevant events occurred, namely the effective sounds and the beeps are relevant events. And what here is kind of depicted how we approach this question. So here you see the EEG or the signal over time. And this is when a relevant event occurs, the blue part. So we compute synchrony here. And of course, we expect somehow that synchrony is high during a relevant event. And then we can have a kind of threshold moving at different levels. And we can determine like true positives, false positives, true negatives, and false negatives for each level of this threshold, and determine an area under the curve of a ROC curve. So in this way, we can see whether we can detect relevant events. And indeed, we could do that. We can do that for the different modalities. And we see again that EDA is sensitive for emotional stimuli, for detecting emotional stimuli. EEG is sensitive for the beeps, for the cognitive stimuli. And we also constructed a multimodal metric, so combining the EEG, EDA, and this is interbeat interval, so that's heart rate, and you can combine those measures as well, and that was is suitable for if you don't know which stimulus you are looking for, basically. Oh, yeah. If you, by the way, have any questions, please go ahead. But just shout because I cannot see everybody. Okay, that was for the lab experiment. We also used data that was collected in the context of this big project to see whether we can observe physiological synchrony in real schools when in students who are recorded using wearables for heart rate and skin conductance. And there were two studies. One study follows 86 high school students for 5.5 hours. There were I'm saying that wrongly. 5.5 students on average were in the same class, and we wanted to kind of use these data to estimate or to see from the physiological synchrony, are the students in the same class or not? So the kind of minimum that we want to see here is that we can indeed detect that people are in the same classroom and being exposed to the same kind of stimuli. And can we indeed do that? And not all students were, so there were 86 students recorded, but they were not all yes, in the same, divided equally in different classes. So there were on average about 5 students in one class. In another study, yeah, there were even a bit less students on average in the same class, and there were other wearables being used. And what we found is that physiological synchrony is indeed higher in the same class than in different classes, between students in the same class than in different classes for heart rate and for EDA. We found that to be significant for the data from study 1. And we found the same trends for study 2, but this was not significant. So these were not enough students, probably, or too small groups. But at least there was some evidence that we could pick this up with these kind of signals. Okay, and I think finally for this project, we looked at focus groups to discuss with students and teachers about the idea of being monitored for attention in school. So that was kind of the use case. But what do they think about that actually? Because yeah, is it useful, or would they not like the idea at all? So it was really interesting to go there. The students really said a lot to us. So here are some quotes. So they said, "We have a right not to pay attention." But also they thought it would be great if this could help to make lessons more interesting, or to help students. And also they were a bit worried about teachers. So apparently then they can all see that we are not paying attention at all, maybe. And also this one I thought was funny. In general, teachers are not great with technology. So it should be a very user-friendly system if you will apply this. Yeah, and so my colleague is actually working on it still. So to divide these different quotes or what they said to different types of ethics, ethical statements. So traditional ethics about privacy, security, consent, ethics of science, Foucauldian ethics. So this was the category that they focused on most, like how does it really change social and power relationships. They are not so worried in general about privacy and security, the students. Okay, so here are some conclusions. So we found that physiological synchrony reliably reflects selective auditory attention in a group, not only for EEG, but also for these peripheral measures, and also using wearables. It was not only this binary classification that we could do, but physiological synchrony captured more subtle differences such that we can predict performance for different modalities are suitable for different types of events. Yeah, I did not discuss this part, but we also did work with unsupervised classification, so not knowing what to because in this in the data that I showed you, we related one participant to one or the other group. But if you don't know what these groups are, what then? So we approached the problem also in a way that we did not know this a priori. And there we also found that a multimodal approach, or combining the different modalities, is useful. and also if you don't know what type of relevant events you are interested in. So there is a benefit to record from different sources of information. First results indicated that physiological synchrony can be detected in real classroom settings using wearables and ongoing conversation about pros and cons of attention detection technology in education. Okay. Oh, wait. So then, good. I shift to another topic. So the part that I told you about was really on attention, where we instructed people to attend to certain elements, or they were in an educational setting. But of course, attention can also be modulated if a task is not specifically on attending, or there is a variation to be expected. So we did also research in mental state monitoring for food industry, and they are not specifically interested in physiological synchrony, but they are interested in attentional processes or processes that are relevant for them. And one of the things that is relevant for them is the food neophobia. Food neophobia is the hesitance to try new foods. And it is a quite general or predictive factor. For instance, we work a lot with Kikkoman soy sauce company. And food neophobia is really predictive on to what extent people buy Kikkoman soy sauce. And so this is interesting for them to understand this pattern. And we did an experiment to use EEG for obtaining insight in early attentional processes when presented with food-related stimuli, the late positive potential, and physiological synchrony. And this is a case where we expect food neophobia to modulate attention. And that is a case where attention is modulated by personal traits. In this case, food neophobia. So our experiment was with 43 participants. They filled out this validated food neophobia scale. We presented them with unfamiliar and familiar food images. So unfamiliar was in this case Japanese food, and familiar was Dutch food, like here, kroketten. And we measured ERPs, so event-related potentials in EEG, locked to these pictures, and we also asked them to judge these stimuli for arousal and valence. And we also showed them a movie on Japanese soy sauce. And that was a 15-minute movie. And for that part, we determined physiological synchrony. Okay, so what we see for the unfamiliar and familiar food images, for valence, so valence is pleasantness, there we see the thing that you would expect. So people with high food neophobia rate unfamiliar food images as less pleasant than people with low food neophobia. And for familiar food images, this is not there. It is food neophobia, right? So only unfamiliar food. This is what is known. And if we look at the ERP responses, so the EEG to these food pictures, we see actually a bit unexpected for us that if we divide the people in food neophobic and food neophilic, so a median split for food neophobia score, we see that the food neophobic people respond strongly to all food images, stronger than the food neophilics. So that is a sign of increased attention. And we had a bit expected maybe to that this was higher really for the unfamiliar foods mostly, but it was for all images. And this was also, if you look at it more continuously, so you look at the amplitude as a function of food neophobia, you see indeed that what I just said, the higher the food neophobia score, the higher the response, both in an unfamiliar case and in a familiar case. And also, if we look at the physiological synchrony during the movie, then we see the same pattern that food neophobia is related to a high physiological synchrony in EEG, indicated of a high attentional engagement. So we found here that physiological synchrony varies with selective attention, not varied by attentional instructions, but modulated due to personal traits, which broadens this potential application of physiological synchrony to monitor attention in natural environments. And for the people in food industry or the food scientists, it is of interest that these results indicate that food neophobia affects short-term attentional processing of all food-related stimuli. So in that sense, it's not really food neophobia, but just food is kind of a stimulus that draws attention. And maybe later on they identify what kind of food it is, and the verbal response is then different for familiar and unfamiliar foods. Okay, I'll just look at time a little bit, but I think I can go ahead. This is this study. We also looked at another way in which attention can be modulated. So we've had this instruction, we had food neophobia, kind of personality trait, and also attention can be modulated by fatigue or sleep deprivation. So we were interested in predicting performance in a sustained attention task when the chances are high that your attention is low because of sleep deprivation. So this was part of a big study where we had participants. We sleep deprived participants one night. So they were in the canteen of TNO here, and we had every time like 8 participants and 2 poor experiment leaders who had to stay awake as well and guard them. And these people, every hour they were asked to watch a 10-minute video. And we recorded again using these same wearables that we used in the experiment that I started telling you about. We recorded EDA and heart rate. And after this video, they performed a psychomotor vigilance task to look at their attention level, like how well they responded to a stimulus, and a subjective sleep skill. So we wanted to predict from this data the subsequent vigilance performance. So first, we pre-tested the movies that we showed them during the night, whether 10 minutes of these movies using this heart rate and EDA is actually potent to elicit physiological synchrony in heart rate and EDA, and that's appeared to be the case. And also it appeared to be the case that again correlated with answering questions correct about the movies. at least for heart rates. And I will skip this. Okay. So the results for here we see the performance of this psychomotor vigilance task, the PVT, and how often. Oh, I did not even explain that task. So people saw a dot, and they had to respond as quickly as possible to that dot. So it's a quite simple reaction time task, actually, but it's quite boring because they look at a screen and just once in a while this dot comes up. And the lapse probability is how often you are not in time. And I forgot what the exact time was. Like if you're not respond within 800 milliseconds or so, you have a lapse. So the lapse probability increases over the night. So this is a very known pattern that around this time, 4 or 5 at night, you are really bad. And interestingly, that does not, and in the morning you get better again, even though you did not sleep. But that is really the circadian rhythm kicking in. The subjective sleeping score is just the longer it takes, the worse it gets, the sleepier they report themselves to be. Okay, so what we saw, if we look now at the physiological synchrony in EDA and heart rate over the course of the night, for heart rate, we did not really see a pattern that kind of matches this, because we kind of were looking for that maybe at this time, when lapse probability is high, physiological synchrony is low. And this y-axis actually is a bit, if you stretch it a bit more, you can see it's a little bit in the direction that we expected it to be, but it's not very clear. But we did see using hierarchical linear regression models that indeed the physiological synchrony in EDA did contribute to predicting the performance in PVT. And for heart rates, it did not work. So it was a modest success, I would say. And of course, the problem that we thought here that it did not work very well is that you really look at different time points. So people were watching this movie that was quite engaging, and then later, okay, they kind of anyway fell asleep. So we have some improvements in mind for future work. Okay, so the conclusions of these two studies are that physiological synchrony in EEG reflects attentional engagement as determined by individuals' personal traits, and we replicated the finding that physiological synchrony can predict performance. Question is correct. And some evidence for physiological synchrony in EDA as predictor of subsequent performance in a vigilance task during a waking night. I think I will very quickly go through this last study because that's of course the nicest study where we looked at monitoring attentional engagement during an inaugural lecture. So I gave my inaugural lecture, and we looked at physiological synchrony in this audience. So where people were really outside the lab in a very noisy environment. So is physiological synchrony above chance in this case? Can we identify engaging events? How do results compare to physical events? And can we distinguish between groups that we expect a priori to differ in the pattern of attentional engagement? So what we did is we recorded 15 people in the category family and friends, and 15 people in the category colleagues. And And here you see a picture of fitting the participants with sensors. So it was, again, the skin conductance sensor. And we had these predefined engaging events. So my brother was processed in slides of the inaugural lecture. And the idea was that, of course, for the family and friends who know him, for them, it is a kind of engaging, meaningful event, while my colleagues don't know him, and that is less of a engaging. Also, people were asked to stand up to see like this body movement. How does that kind of compare to really mental events? We had a kind of workload task and a stress task. That was instruction to sing a song. We had a video of my son, who again was more meaningful or emotional probably for my family and friends than my colleagues. And another engaging event. So we found that indeed physiological synchrony was above chance for almost all participants. And also we could identify these predefined engaging events. And I think this is, yeah, so the standing up event was in about the same effect as the mental events. And can we distinguish between the groups that are expected to differ? Yeah, these are not the latest results, but we could find that difference also. And here's my last slide. From physiological synchrony from lab to real life. So I started this talk also by lining out the challenges that we have to do this mental state monitoring in real life. So one of the challenges is that we need labeled training data. If you make machine learning models to track attention, you need to train your model on labeled training data. But here with this kind of feature, you don't need those training data, and you kind of circumvent the ground truth problem. That is a problem if you want to label training data, like what is attended to and what not, for instance. And also we circumvent context specificity. We also found that wearable sensors are informative, which is good for real life applications. So directions of applications are evaluating individuals' engagements with a lecture or meeting, implicitly and continuously over time, detecting relevant events so we can kind of find out what do people think is relevant, and what do novices maybe think is relevant, or experts in certain area. We may be able to detect some oddball, like maybe one student is not paying attention or what is going on there. Maybe, and this could be helpful in applications like online education, where the teacher doesn't have alternative information about attention level in students. And I want to thank these people, especially Ivo Stoeldreyer, who is a PhD student who recently, the start of this year, defended and obtained his PhD on this work of physiological synchrony. Thank you.